Hello students, I am going to explain about the topic of gene interactions. Before we are entering into the topic, I would like to explain about what is gene. Gene, it is made up of DNA. It is having the hereditary material. That hereditary material, it is carried forward to the next generation. Here you can see the definition for gene interactions. Two or more genes will be affected and expressed in a single character of a particular organism. It is known as the gene interactions. Based on that gene interaction, it is divided into two types. One is interallelic gene interaction, another one is intra-allelic gene interactions. Here they are using one new term, that is for allelic. What is that allelic? That is nothing but two or more alternative form of gene that arise by mutations. It is sudden changes. It is occurs in the DNA nucleotide bases or found at a same place on organisms, that same place on a chromosome. It is known as allelic. Here I would like to explain in detail in the types of interallelic gene interactions. Yes, that interallelic gene interaction, it is nothing but allele of one gene with the allele of other gene are referred to as interallelic gene interactions. So this interallelic gene interaction is also divided into four major types. First one is complementary gene interaction, another one is supplementary gene interactions, third one is epistasis. That epistasis is also subdivided into two types. One is dominant epistasis, another one is recessive epistasis, and finally non-epistasis is also comes under for interallelic gene interactions. And the experimentally they are proving that the gene interactions, how it is coming for F1 and F2 generations. They are taken into the examples and various sources, and that parental genome is also taken here. That everything will be discussed in that table for interallelic gene interactions. Here I would like to explain in detail note on complementary gene interactions. For that example, they are taken into the flower color in Latharus odaris plant. Yes, what is complementary gene? That complementary gene, that complementations between two non-allelic genes are essential for producing a special phenotype that is called as complementary factor or complementary gene. What is the special phenotypes? The phenotypes, we can observe the characteristics of an organisms like hair color and shape and size of an particular organisms, that is noted here in that F1 generation or F2 generations. Like that, the second point they are discussing about that the two genes are involved in a specific pathway and they are producing the functional products are required for gene expression. Then one recessive gene, recessive allelic pairs are produces that in that changes interaction, gene interaction, they are producing that the mutant phenotype of their offspring. Like that, the complementary genes were crossed together, that here they are taken into the dominance alleles are present together in that complementing each other to produces the yield of complementary factor on that respective organisms. It is resulting to produces the new special phenotypes characters. When that new special character are produces in, we can observe only in the offspring, the next generations. Likewise, the gene loci have homozygous recessive, that same locus, it is identical, it is each other, no? That a homozygous recessive alleles, they produces the identical phenotype in their F1 generations. You can see it in the changes into F2 generations, and the respective ratio is 9:7. How it is coming that 9:7 the ratio, it is complementary gene. We will see it in the coming slides. I will explain it to detail. Here you can clearly view that the examples of complementary gene. Here the example of Latharus odaris plants, the plants flower color, one is blue, another one is white. The two variety of plants are coming from same plants, that Latharus odaris plants. So because of gene mutations, gene variation, gene interactions, it is produces the two variety of flowers in a single plants. That is, we can view here in plant, that sweet pea plants, two variety of flower, white flowering plants were seen here. That white color with that original color is white flowerless, but because of the gene interaction, it is produces the blue color flower in sweet pea plants. So according to the Patterson and Punnett, when two white variety sweet plants were crossed together, we can observe in that changes in offspring. It is produces the new character, that is for purple color flower in F1 generations. Likewise, the F2 generation is also produces the changes in the gene mutations occurs in the F2 generation is also we observed here for 9 were purple color plant and 7 white colorless sweet pea plants. Likewise, it is produces the inheritance of gene interactions with each other for complementary gene. So it is complementing each other. With inheritance of flower color sweet pea plants, how it is crossing each other, how it is producing that special character on the phenotype of respective plants. We will see it in clearly here based on the Patterson and Punnett. That variety 1 white colorless plant and variety 2 is also taken that white colorless sweet pea flowers. The parental genotype here they are taken into the small c, capital P and capital P. Likewise, the variety 2 it is taken into the capital P and small p, capital C and small p and small p. Here they are denoting the gene symbol like this. So I would like to explain that symbol that taken here, the small, that upper cases, that is lower cases and upper cases, they are using that symbol to cross each other. The genetic variations in we can observe into F1 and F2 generations. No, so like that the parental genome that is noted here for the lower cases of small c and small c for recessive alleles, and capital P and capital P, that is upper cases, is also denoted that the dominant genes. Likewise, it is the two variety of flower color, that white flower will be taken into the example of this complementary gene interactions, that again the parental genotypes are crossed together, they should produces the gametes on that. Because of that gametes, it is produces the purple color and the flower. So before we are seeing that two variety of white colorless sweet pea plants, and now we saw that the F1 generations we got the result in colored, that purple colored sweet pea plant. Because of that gene will be inherited in that crossings. So you can see the F1 generations. Likewise, for F2 generation is also having the different variety will be produces. Yes, how it is produces, yes, we can see it in this slide. F1 generations, that is F1 generations we got a purple color flower. That F1 generations purple color flower, it is self crossed. It is also producing the F1 gametes. That F1 gamete, it is crossed together. That gametes are arranged in a manner like this. So once you crossed with each other, the F1 gametes, it's got produces the 9 purple color and 7 white colorless plants we got in the next generations for the F2 generations. So like that they are crossing the Patterson and Punnett. They are proved that for changes occurs in the gene for Latharus odoratus plants, F2 phenotypic ratio is respectively 9:7. So I hope you people are understood very well about the concept of complementary gene interactions. Thank you so much to watching me in this video. Thank you all.